with sensors, we've been talking about different kinds of individual responses that we could make, whether in the presence or absence of a particular molecule. If we revisit, for example, the lac operon, we know, as is shown here, that it's a natural sensor for two different kinds of inputs, whether glucose is around or not, and whether lactose is around or not. And so, particularly when we've got two different kinds of inputs, think of, well, I guess when you've got, it's perhaps more about the fact that you have two different states, on or off, but when you have two different inputs as well, two or more, you start to imagine that you don't have a simple sensor, you have something that's a little bit more like a logical operation, a circuit. And so what's kind of shown here is a representation in zeros and ones that starts to bridge this idea of the logic that exists in some natural genetic regulatory networks with concepts from electrical engineering and computing, where binary code is very common. And so you can then actually imagine this as more of what's called in that field a truth table, where these are your inputs and this column here is your output. And your output tells you, are the genes on or off? So the one here means, under this condition, and in this truth table, under this condition only, expression of catabolic genes is essentially true, only when lactose is there and when glucose is not. So this is a particular kind of circuit, and you can maybe see that more clearly here when you view this as the different kinds of digital logic gates that come from computing and electrical engineering, because they are the basis. Think of them as the simple machines equivalent, but for a circuit rather than some kind of hardware, some kind of machine that's made up of simple machines. Your homework is going to dabble into this, I think, a little bit. But what I want you to see is that there are these different kinds of logic gates that are used in other fields. And the notation here on the left column, you've got the gates are often called. Some of this may be familiar to some of you, if you, depending on what kind of other courses you've had. If not, that's okay. There are symbols shown here. I personally don't think that we need to concern ourselves too much with the symbols, although these are simplified representations and they're standardized in the field of circuit design, at least for what kind of operation these are. We also don't necessarily need to worry too much about the notations, but I would think about things in terms of a truth table, because as you can see in 5 out of these 6 cases, you've got 2 inputs, an A or a B. You only don't have that here in the NOT gate. And this example of a NOT was actually what we just saw, the inverter with the ATC and GLK example. And so if we're thinking about the lac operon, I don't think that example is actually one of these standard gates. So some systems that have developed biologically, like in this case, I'm only going to turn something on if there's one and not the other, but it has to be just this one. That's not a standard operation that you see in computing at the level of one gate, but it's entirely possible that through the coupling of some of these different gates, you can get that output. And in fact, that's how circuits are built. And I think your homework goes a little bit more into that. And so this is, just an example here shown, not for us to actually discuss, but, in other fields, for example, one might have to figure out, okay, you've got these different inputs, A, B, C, and you've got these symbols here, and these are the ways in which they connect. So what's the truth table? What's your output going to be? And you can also simplify some of these. And these are important if you're trying to build a device from the programming perspective, because just like with code, you might not need to write out something repetitive in every line. One or two lines with a simple for loop might accomplish the job for you. So I wanted to kind of give you at least one illustration of not necessarily why this might be the most useful, but I just thought it was an interesting example that can hopefully help you think about cells as maybe more than just devices that produce a particular metabolic product in high quantity, right? We already know that some of us already think about cells that way. If you come from the human biology perspective, you know that our cells are more interesting and complicated than that. And in fact, someone who I believe is a co-author on this paper and from the lab of Chris Voigt, who does a lot of this genetic circuit design work, really pioneered that field, once told me that the reason why we think about engineering genetic circuits and particularly synthetic biological circuits is because it basically elucidates for us the process of human development. And it took me some time to kind of think about that, because most of these examples are in bacteria, and so I'm not really thinking about human development, but it's the same underlying principles, right? When your cells have to decide, do I become a skin cell or brain cell or whatever, there's this myriad of factors that are being sensed and then being processed accordingly. Your cells are performing incredible feats, not just during human developmental biology, but even in adult stages. And so sometimes this can be easier to pictorially represent than in other cases. And so I thought this example of in computing a simple calculator display was a relatively good one for at least illustrating the power of this kind of approach. So you can imagine that you have 7 different wells here, A through G. So A, B, C, D, E, F, G. And you're going to have 4 different inputs. And so your different inputs are like your different inducer molecules. And then you've got circuits that are shown in this overlay here, because that's just often how the field of circuit design works. But really, you could trace back and you have one particular circuit for each well. So imagine for A, you've got a particular strain of E. coli that's engineered with one circuit, such that A is going to always be the same set of cells across the board here, but then you're going to give it a different combination of these 4 inducers. Like you got these 4 inducer molecules, and maybe in this first case, for example, you're not going to provide any of those molecules to that well. And the same for the rest of these. And so this is, in effect, your truth table here. And this is what happened when researchers did this experiment. And so again, it's meant to be, I think, a cute visualization of how, if you imagine again this strain in well A that's been engineered deliberately to give you this kind of profile. We can see that, in some cases it's brighter than in other cases, but it does that with relatively good fidelity. And likewise, when we do that for B, C, D, E, and F and G respectively, you can clearly tell here what number you have. And so thinking about actual sensors, say, if you have a device where you want to sense some molecule maybe coming out of a CHO cell fermentation, you want to look for maybe 4 different molecules, and so this is right where the real value comes. If you actually care about 4 different molecules, you want to be able to tell all these different combinations, you've got a device here that gives you a different number based on the combination that you have, and it's pretty clear. I mean, maybe in this 5 and 6 case you have a hard time telling which one you have, but now you've got an interesting, pretty innovative kind of readout. And so this is just one example to highlight the power of why these biosensors, when assembled together in different ways, in addition to enabling strategies like dynamic metabolic control, really give you, and the field of metabolic engineering, new abilities. So with that, I think I have, touted biosensors and synthetic circuit design as much as I reasonably could. And that kind of concludes our unit on gene regulation, expression engineering, and biosensing and circuit design. So this was a new unit, piloted out for the first time, not part of this course ordinarily. And I scraped together this material, not quite before every lecture, but just in the past few weeks. So appreciate any feedback you have on it, and hopefully you found it useful. We're now going to go into a short other unit that continues our progression from thinking about individual enzymes to broadening our aperture to pathways and then to gene regulation. And those networks are usually bigger than a pathway, or at least I tend to think of it that way. And circuits were part of that. And they could be small at the sensor level, they could be big. But now we're gonna think about the whole genome, or at least strategies to enact a lot of these changes. And so that's what we're gonna get to. Specifically, if we wanna make changes to the genome, first, how do we make those changes? Then we can talk about what kind of changes we want to make. And we've talked about, things like knockouts and upregulation and downregulation a little bit before, but we might dive into other kinds of genome engineering that are possible, some of which are helpful from a metabolic engineering perspective, some of which may have to do with protein engineering a little bit more, or with questions of fundamental to life, like the minimal life, or the quest for it. So that's where we're going next.